So how does the nephron handle sodium? Now this is where I deviate from Guyton and favor BRS and Costanzo, in the sense that BRS basically deals tubular function according to ions or according to the substance. It makes more sense. Uh, it's more holistic. Uh, as you can see, he is shown the entire nephron, and he just discusses sodium in this particular diagram. Um, and then there are diagrams for hydrogen, there are diagrams for bicarbonate, for potassium and calcium, and so on and so forth. So, in my experience, this promotes learning and retention of the tubular function better than what Guyton does. And what Guyton does is he discusses it segment wise. So he will pick uh, proximal convoluted tubule and discuss everything that is happening in this very busy place. Then next up, he will take up loop of Henle and then DCT and then collecting duct and discuss individually, i.e. region wise. In this, I've seen that there are problems because when you talk about, say, ECF volume handling by the nephron, you're basically interested in sodium. You're not interested in, say, for example, how does hydrogen get sorted out at PCT and DCT and distal collecting tubule, convoluted tubule, and so on. You're only worried about sodium in this particular scenario. And then acid base, you're not really concerned about sodium. You just want to know about hydrogen. This consolidated view is given extremely well in BRS, okay? And hence my sequence of lectures will follow the substance specific handling of nephron, okay? So I deviate now from Guyton, not region wise. I will be looking at things from the substance point of view. So sodium handling by the nephron is being shown to you here, and you can see that proximal convoluted tubule, maximum, the largest share of sodium reabsorption takes place here, 67%. Then you have thick ascending limb of loop of Henle, in which there is 25% reabsorbed, sodium is reabsorbed over here, and in between these two, there's about 8% of sodium which can be, and this is important, which may or may not be reabsorbed from here, and less than 1% is excreted, normally speaking. Now this again is a normal situation. We're not talking about any volume contraction or expansion. This is just normal. Another point to note is that 67 and this 25% is fixed. Okay, this is going to happen no matter what, generally speaking, unless they are at a constant GFR, of course. Uh, and constant GFR means the flow velocity is normal as well. 67% and 25% is constant. The variability, it comes here at 5% at DCT and 3% at the collecting duct. Variability, why? Because of this reabsorption here in this region is affected by aldosterone. Okay, aldosterone, as we have mentioned in bits, is the sodium reabsorption hormone released by adrenal gland, and it's one of the main stimulus of aldosterone is, you know this, it's angiotensin 2. Okay, this is a good diagram to know the overall handling of sodium. Now, if you could delve into some specific details uh, briefly, this of course does not replace your reading of Guyton, and you can always read it region-wise as well to enhance the depth of your knowledge. So sodium here is shown. I beg your pardon. PCT cell is shown. This is the proximal tubule, early cell PCT, and this is the late proximal tubule cell. Okay, there are differences. We will look at that. So this is the lumen. This is the luminal border of the cell, and this is the basolateral border of the cell. Now remember one trick here. Always remember that sodium potassium ATPase at the basolateral border is abundantly found along all the length of the tubule, whether it's PCT or elsewhere. This ATPase is there just about everywhere. Okay, and you know what this ATPase does. It throws out three sodiums and bring in two potassiums. So it constantly creates a deficiency or a gradient for sodium. Okay, so sodium concentration in the cell, said another way, is less, is kept at a lower end because of the constant working of sodium potassium ATPase. Now, this is an ingenious method in the reabsorption capital of the tubule, which is the PCT. So see how sodium is then manipulated to do all sorts of things, okay? Whether it's sodium glucose co-transport, sodium amino acid co-transport, sodium along with various anion co-transport, or sodium and hydrogen antiport transport. Sodium concentration gradient, which is created by sodium potassium ATPase, is coupled to do so much, quantitatively so much at the PCT. Okay, so these are those famous symport mechanisms which bring in the stuff like glucose, amino acid, and so on into the cell by coupled transport, and then there are dedicated transporters for each one of them at the basolateral border, which brings it to the interstitium where it's picked by, not shown here, but it's picked by the peritubular capillary network, and off it goes to the main blood. So you reclaim it, you reabsorb the whole, the crucial things. Okay. So in this, there is important. Yes, we've done this. The one difference. Let's just do this first, and then we'll do this. One of the differences, very main differences, if you can spot it, between these two cells is, look at this cell, look at what is being transported at the apical or the luminal border. Then compare it here. See any difference? Which is the anion which is being transported within the early cell and then the late cell. This, look at this. It's bicarbonate, basically, which is being transported or reabsorbed here, and it's chloride in this case. Okay, so sodium is being dealt here, not chloride. This is one of the differences. However, in the late cell of the PCT, chloride is being also dealt with along with sodium. Okay, so this is one of the differences. The proximal cells don't tend to deal with chloride, but the later cells do. Okay, this is one thing. Now coming to this part. So what does isoosmotic reabsorption means? Isoosmotic reabsorption is very crucial. It's very important to understand in this context what is happening. At the PCT, remember this point please, it's very, very important. Equal part of sodium is picked up along with equal part of water, isoosmotic. So from the lumen, if there is one ion of sodium which is transported inside the cell and then eventually to the blood, another molecule of water will accompany this sodium. Okay, so you don't, the fluid basically which enters the cell and then which enters the blood has the same osmolarity. There is no difference between water to sodium ratio, if you know what I mean. Okay, this this PCT phenomena is called isoosmotic reabsorption. Now there are very important implications for this. So when the filtrate, fresh filtrate from the glomerulus entered the lumen here in the PCT, it had a osmolarity of around 300 milliosmol per liter, right? We remember the plasma osmolarity, and since it's literally part of plasma that has filtered out at the glomerulus and now has entered the lumen in the PCT, it is reflective of plasma. It was plasma before it got filtered out, so the osmolarity will be 300. Now, the volume, the osmolarity of this fluid when it exits the PCT and enters the loop of Henle system, the osmolarity is still 300. The volume has decreased. Very important to note. The volume has decreased because you have reabsorbed so many things from it. However, the osmolarity is the same because of this phenomena that I've just explained. Sodium and water have been reabsorbed together. So this is called isoosmotic reabsorption. Okay, it's very important later on for us to differentiate between this type of reabsorption with what happens later on in the loop of Henle, especially in the loop of Henle, and then in the DCT as well. Okay, so this is an important point to note. Another important point to remember is, remember angiotensin 2. We have not done this particular function of angiotensin 2, but right here at this point, we need to mention that this counter transporter, which is very crucial for acid base topic, it gets stimulated by angiotensin 2. Its broader understanding, its broader application will come when we'll discuss acid base. But suffice to say is that any uh, issue with volume will trigger angiotensin 2. Any issue with ECF volume should affect angiotensin 2. If you do not understand this concept that I've just mentioned, ECF volume's relationship with angiotensin 2, please pause this lecture and go back to the lecture which describes angiotensin 2 in part of when we discuss the TG balance, the tubular glomerular feedback mechanism under GFR autoregulation. Okay, I'll try to put a link here so that you remember. If you do not understand how ECF volume is linked with angiotensin 2, it's not a good sign. You need to stop this lecture, go back, do that, and then come back to this part of the lecture. All right. So for those who follow, angiotensin 2, once it's available in abundance, it triggers sodium hydrogen antiport mechanism performance to a higher grade. So it is normally working fine, but when angiotensin 2 is available in abundance, it triggers it. It enhances its function. So the perceptive student will quickly see hydrogen being more secreted in this scenario. Yes. So you have a deficiency of ECF volume, which triggered the angiotensin 2 system. Angiotensin 2, when it came to play uh, at a higher concentration, enhanced sodium hydrogen antiport mechanism, which basically meant sodium was absorbed more to address the decreased ECF volume. But at what expense? The expense of hydrogen. Hydrogen was excreted out. Okay, so this is called contraction alkalosis. Contraction means volume contraction. ECF volume was contracted, and to address that, this antiport mechanism was triggered. Hydrogen was lost more than usual, which led to alkalosis in the body fluids. Contraction alkalosis. Remember this. This is an important question, especially with good students in the Viva. All right. Okay, this is, don't worry, there might be too much alphabets on this slide, but it's actually very nice and simple. Uh, Forget this is the summary of what I'm going to explain right now. So this here is shown a PCT cell, and isoosmotic reabsorption is taking place. Everything is honky-dory. ECF volume is fine. There is no decrease or increase in ECF volume. That filtration fraction at the glomerulus is coming out nicely at 20%. No problems in GFR. Everything is fine. The tubular capillary is fine. The status of the PCT is fine. So filtrate coming in the lumen, it gets reabsorbed isoosmotically, which means every sodium is accompanied by water, and it comes through here and through here. Now remember, and this is where the new information is coming through. When you remove plasma as part of the GFR process normally from the blood at the glomerulus, the rest of the blood is slightly thicker, isn't it? So afferent, imagine, afferent arteriole brings in whole blood. Then glomerular capillaries filter out 20% of that blood's plasma. and the filtrate is formed, which is transported into the PCT. The remaining blood minus the 20% of plasma, which is now become slightly thicker, it exits the efferent arteriole and now becomes part of the peritubular capillary network. And now it comes right next to the cell which is dealing with its lost cousin, the plasma in the lumen. So imagine that the blood got split, 20% got split at glomerulus and came here, right here, okay, in front of the proximal coronary tubule. The rest of the blood, minus that 20%, through the efferent arteriole, it went through the peritubular capillary, okay? And in the peritubular capillary, now it comes here, which basically is sort of in front of that filtrate that got formed a few seconds ago. So this relationship is called GT balance, glomerulo tubular balance. So what the glomerulus did, which resulted in formation of the fluid, that with whatever now the tubule will do with it, glomerulotubular balance. Okay, please do not confuse it with the tubuloglomerular feedback mechanism, which is part of the autoregulation. Okay, tubuloglomerular feedback is all that renin, angiotensin 2 stuff that is separate. Okay, GT balance is a proximal convoluted tubule concept. Okay, and what is it? So in isoosmotic conditions, when the lumen got its fresh filtrate, and you will now remember we said 20% is filtration fraction, and most of it is reabsorbed. So most of the reabsorption takes place at the PCT, as you can see by these dotted lines. And here, the peritubular capillary. What is this? This is the plasma colloid osmotic pressure. Now, since you took out plasma, what was left is thicker. When I say thicker blood, what I mean is that the concentration of plasma proteins and all those molecules which affect osmolarity, they increased. So the plasma osmolarity of this fluid will be slightly increased, and the oncotic pressure is basically the stalling force which is relevant to this. When this type of fluid, blood comes in here, and all sorts of reabsorption is taking place, you can now predict what this slightly increased oncotic pressure will do to the incoming reabsorbed fluid. It will attract it, right? It will attract it. If you do not understand this, please stop this lecture, go back to the stalling forces discussion, which we were discussing early on, and then come back. You can come back to this lecture. Okay. So increase oncotic pressure will attract the fluid which is coming in the interstitium due to all those maneuvers that we've discussed in the previous slide. And this is how you save the day. You do your isoosmotic reabsorption, no problems. Now there are two scenarios which are different. One is volume expansion. So something has happened in the ECF. Okay, it's not the kidney's fault. Something has happened in the ECF and the volume has expanded. And the second scenario is the opposite. Something has happened in the ECF which has the person has lost volume. So the volume has contracted. See how GT balance saves the day in this scenario. So when the volume was expanded, you do your normal GFR, filtrate comes here, but check the status of the blood now. The peritubular blood has higher hydrostatic pressure. Okay, it has higher hydrostatic pressure and decrease oncotic pressure. Okay, again, if you don't understand how this happened, please refer back to the stalling forces discussion in the earlier lectures and come back to this. So this increased hydrostatic pressure will retard the reabsorptive fluids which is being reabsorbed by the PCT. Okay, and it makes sense because you don't want extra volume because you already are extra. Okay, once again, ECF volume has increased in this scenario. Okay, and now the kidney is filtrating it, and now the blood that is coming in has higher hydrostatic pressure than normal because of whatever you did with that ECF which has increased its volume. Okay, this is being expressed as the increased volume in the ECF is being expressed as increased hydrostatic pressure in the peritubular blood. Okay, this will uh, not facilitate too much reabsorptive fluids to enter into the blood, and hence that fluid will be, i.e., discouraged to come to this side. It will remain in this side, form extra urine, and is excreted by the body uh, in its attempt to address the expansion. So if you start excreting extra fluid in the urine, you are decreasing ECF volume back to normal. I hope you understood that. This is just the opposite of this. So I'm not going to explain the whole thing. You can just apply the same understanding. Just remember that in this scenario of volume contraction, what has happened is that the hydrostatic pressure has decreased, the oncotic pressure has gone up. This will increase reabsorption, and uh, uh, less amount of urine will be formed, and everyone, the ECF volume will go back to normal, and everything will be happy. This is just a summary of what I've just described. Okay, enough about PCT. Please do give it a formal read from your textbook, and things should settle down. Now, at LOH, loop of Henle, we are not very much concerned about the thin descending limb. We are basically, and not the thin ascending limb, we are basically concerned with the thick ascending limb. Those thin segments basically deal with water and other stuff, less with sodium, but it's the thick ascending limb of loop of Henle which basically is shown here, this. So this thin descending and thin ascending is doesn't do much in terms of sodium. It's the thick ascending limb which basically deals with 25%. So there's a significant amount of sodium that gets picked up here. So this is that amount, and what happens in this section is what we are discussing here. So you can see that there is a big symporter on the luminal surface, which basically takes one sodium, two chloride, and one potassium in one go. So all four molecules of ions from the lumen into the cell, while the potassium is secreted. The sodium and chloride are basically reabsorbed to the interstitium. Potassium can be secreted and is also reabsorbed here. So that's one thing. The main point here in this segment is that this symporter plays a very important part in concentration of urine scenario. Remember this. We'll be discussing this under water homeostasis. This symport mechanism is crucial in concentration of urine. The second point to note here is that sodium is reabsorbed without water. So this is the single effect. This segment of the nephron, the thick ascending limb of loop of Henle, this is where the single effect takes place. Sodium is reabsorbed, but water is not allowed to follow it. And the reason it's and the way it's done is that cells here are not permeable to water. This is indeed, this segment of the nephron is called the diluting segment. Basically, solutes are removed from it so that the filtrate becomes dilute. It's called the diluting segment. Okay, and even the paracellular pathways, the tight junctions between cells, are so tight that they don't allow water to come in. While you are basically separating sodium from its buddy water in this segment. This is the single effect. Okay, this is another very important point about this segment, and the sodium transport here is peculiar in this sense. Please note this. Um, the third point is ADH, antidiuretic hormone, stimulates this transport. Again, ADH will come in very heavily when we talk about concentrating urine in water homeostasis. So we will talk about this in detail over there, but this is the third point which is important. The fourth is this thing called, which is written here, it's furosemide. It's a diuretic. It's a drug which is used to increase urine formation, especially in patients who are suffering from hypertension or heart disease, so that their ECF volume is not overburdened, and hence they don't have high blood pressures. So these type of drugs are given to them, furosemide, which basically blocks this channel, all this system. It blocks it. So these ions are not reabsorbed. They just go from thick ascending limb loop of Henle to DCT and collecting duct. Very importantly, along with the fluids, and hence they are excreted of the body, taking all that volume with them, which is important. Okay, in patients of hypertension. So furosemide is a diuretic. Remember that. This is the fourth point. Okay. Okay. So we now move on. This slide shows you two cells. Okay, so this is, we are at the DCT now, the distal convoluted tubule. You know, the place where sodium is reabsorbed variably under the influence of aldosterone. So the presence of aldosterone or the absence of it will make all the difference. And you should remember that aldosterone is under the influence of angiotensin 2. And you should remember when angiotensin 2 is released. Again, if you do not understand the link between ECF volume, GFR, angiotensin 2, and aldosterone, you are referred back to the lecture that discusses tubular-glomerular feedback under GFR regulation. Please go and first try to understand that and then come back to the slide. So we are in the DCT. Just to give you the reference, this is where we are. This is where we are. The whole DC segment, which then goes into the collecting duct. So there will be early cells. We call them the early DCT cells, and then the later part of the DCT. There is a slight differentiation. This whole segment basically has two types of specialized cells: the principal cells and the alpha intercalated cells. Okay, once again, there are specialized cells here, which are called principal cells, found only here, and alpha intercalated cells. Okay, alpha intercalated cells are to do with potassium and hydrogen, and hence will not be today's topic. However, the principal cell is the prime sodium dealer of this segment, and it basically corresponds with aldosterone. Aldosterone's target cell is principal cell. Okay, so hence we will be talking about principal cell and what is it and what's going on. So this basically is a principal cell. But before I come to this, in the early DCT, cell of the early DCT, you can see that it also deals with sodium and chloride, and eventually chloride is reabsorbed here, and sodium, you know what's happening here, the ATPase. The important thing is this co-transport is basically can be blocked by another class of diuretics. So we discuss furosemide, that is at the level of the thick ascending limb of loop of Henle, while thiazide diuretic basically is early DCT cells blocking this. And if you block it, what will happen is that sodium won't be reabsorbed. Sodium stays in the lumen along with its water, and it's drained out by urine, taking along its water, and hence the extra volume that the body gets rid of, the diuretic effect, helps with the ECF overburden of volume. Again, this diuretic is used in hypertensives to reduce their blood pressure. You really need to be okay with the volume pressure relationship. All right, this is the mainstay of one of the prime functions of the kidney. Now coming to the principal cell. Okay, a word of caution: all these diagrams, the blue ones, the blue boxes, they are from BRS, and hence the lumen it's just a little detail. The lumen is shown on the left, while the basolateral border is shown on the right. This is from Guyton. In this, the lumen is shown on right hand side, and the basolateral border is on the left hand side. Just a little caution here. Okay, so what's happening? What do you see in the principal cell here? This is the principal cell. Okay, so you see that the sodium potassium ATPase at the basolateral border, it's doing its business, keeping sodium concentration down within the cell. This is then manipulated by the apical side, the apical border, and sodium is being reabsorbed. Now, something else is happening. Sodium is being reabsorbed and potassium is being secreted, something to note. So principal cells reabsorb sodium and they secrete potassium, okay? And this whole thing is stimulated by aldosterone. So principal cells of the DCT, when they are acted upon by aldosterone, they increase their sodium reabsorption, and as a consequence, they increase the potassium secretion. This has very important implication. So sodium gets reabsorbed, and sodium is linked with ECF volume. I keep on repeating this so that it solidifies in your mind. But it's also coupled with potassium secretion. And potassium, you know, is a crucial ion for heart and all the other matters. So basically, what is happening is sodium reabsorption is being linked with potassium excretion. Okay, this is an important component, and it will be tested in various ways. So for example, if there is a tumor of the adrenal gland which is secreting abnormally high aldosterone levels, now this has all sorts of implications in this patient for sodium and potassium homeostasis. Now you understand what's going on. Okay, so this person will abnormally reabsorb sodium and, very importantly, get rid of extra potassium and then go into all sorts of cardiac abnormalities. Yes, so here is an example of an endocrine disorder messing up a kidney function, and it's linked to a heart function problem as well. So now you understand the complexity which is related with this principal cell. Okay, um, that's one thing. Okay, so there are sodium channel blockers. Again, these are diuretics which act on the channel side. They block it and do the same thing which I've discussed that diuretics do: increase urine volume. Then there are other um, diuretic class of diuretics which act on aldosterone. So they compete with aldosterone to bind to the receptor which binds to aldosterone, and hence they don't let aldosterone do what it does, which is sodium reabsorption and the attendant water reabsorption. Again, that will have a diuretic effect. All these are diuretics. It's just the site of their action is different. Sodium channel blockers literally block the channel itself, while aldosterone antagonists compete with aldosterone so that it doesn't do its job. Both the result of both is the same. A footnote about this is that this, by the way, is also a cell which is involved in water reabsorption via the ADH. So you can imagine it's a very busy cell. It's reabsorbing sodium, it's secreting potassium under the influence of aldosterone, while it can reabsorb water when ADH is present.